Thanks for the introduction, and thank you for having me here. It's a pleasure to speak today. All right, well, so here we are. There is no magic bullet is the title of the talk, and we're going to talk a little bit about COVID-19. Fractal Therapeutics' background is not COVID-19. It may surprise you to know that we were doing nothing on COVID-19 before March of this year. So when the pandemic hit, we pivoted towards COVID-19 on the premise that at least, as shareholders in sort of the in our common human fate, there were some questions to be asked around what could we be doing? How bad is this problem? How bad will the problem get? So what I'm going to talk to you about today is really what we've been able to do and what we've learned so far. We actually have active therapeutic programs for COVID-19. I'm not going to touch on those today, but this is very much a pre-competitive talk, again, sort of exercising our stake as our shared humanity, and really kind of putting it out there, things that the biomedical community could and should be thinking about for COVID-19 as the year goes by and as the pandemic continues to develop. So, it gets said very often, it's kind of like a cliche, the defenders are down to their last bullet and, here comes the cavalry, right? And if you've been paying attention to the way things have been going with COVID, not just in the US, but also in Europe and worldwide, things look really bad right now. And there's this real sense of optimism around the biomedical interventions and, things are just around the corner. We're about to turn a corner on this and so on. And it's been said, the New York Times had an article where they used the term, the cavalry is coming. And this sort of trope of the cavalry coming to rescue us has been actually verbalized on multiple different occasions. And so that's a really tempting way to think about where we are right now. And unfortunately, it's my sad duty to inform you that that might not be the best way to think about things. Optimism, especially unwarranted optimism, is a bad basis for strategy. And this virus is much tougher than we all had anticipated in the spring. There's probably not a person alive today who could have guessed that we would be here. A lot of people in the spring said, it's not the flu. Now people are saying, it's not the Spanish flu. And a case can be made that the Spanish flu might very well be a best case outcome for us at this point. And the reasons for this is it's a coronavirus. It's a cold virus. It's like the common cold. And we know this intuitively, but we haven't sort of fully embraced that. And we're still thinking about it in terms of the flu. So today I'm going to show you work that we've done around some of these topics. So we have a slew of papers coming out right now. I'm not explicitly referencing work from the literature, but there's a lot of work from the literature that can be tied into this and is tied into this. What we're finding is in lockstep with what a lot of the literature is saying. And some key points are immunity against this virus is short-lived. Natural herd immunity alone is not practical because immunity is short-lived. Noncompliance will be a much bigger problem than people anticipate because even a small percentage of noncompliance will really grind us to a halt in terms of eradication. And a modestly effective vaccine alone will not hack it. However, stacked interventions might work. And we'll have to think about, when we think about therapeutics, when we think about prophylactics, everyone says, don't worry about mutations. I'm here to tell you, worry about mutations, and I'll explain why as we get into the talk. So one of the things that we do, so Fractal Therapeutics, we're basically a drug discovery and development shop, but we're not too proud to learn new techniques. And getting into this pandemic, the first new technique that we picked up and embraced was epidemiological modeling. Many of you know this kind of modeling. It's often done to predict the future. You have that sort of laughable model that IHME predicts that every 3 weeks they make a prediction and then every 3 weeks they re-update their prediction quietly because they got it wrong. So epidemiological modeling can sometimes get a bad name, but it's most useful not as a tool for predicting the future, but for driving strategy. Epidemiological modeling is really useful when you ask a question of, okay, well, what would happen if we did this, or what would happen if we did that? That's where it's the most useful. It's the most useless when we try to guess what people will do. So what we do is we use epidemiological modeling to map strategy to outcome, rather than using epidemiological modeling coupled with wild guesses about what people will do to figure out what the future holds for us. So this is a standard epidemiological model. What you have is a susceptible population in blue, S. Some of them get exposed at a certain rate. The exposed folks either get infected, they get infected at a certain rate, and then they recover. Now, the modeling that I'm going to show you next shows you the impact of quarantining or symptomatic isolation. And in that case, in this particular model, we had a subpopulation that was symptomatically isolated. Not every model will have that, but you can design different kinds of models to address different kinds of questions. And it's a super practical approach, very similar to PK/PD modeling in terms of how it works, that can be used to ask questions about what happens. So we used a model like this, and you can see sort of the time course over a hypothetical epidemic in that panel C over there, which shows you sort of what happens in a population when you don't do anything. Over time, the susceptible population decreases, the exposed population increases at a certain rate. Some subset of those people become infected, and those people eventually go on to recover. So now we used models like this to ask strategy questions. So the first question we asked was, what happens if you isolate people symptomatically? And we asked the question, if you look at the number of days after symptomatic onset versus the percentage of population removed, what happens to the disease control? And these heat maps, let's focus on the one on the top right panel, which is what happens if transmission is at 150% of what it is today. And then we look at the percentage of people infected. And we look at quarantining strategies that involve, well, how many days after symptom infection, after symptom development do you need to be quarantined? And what percentage of the population do you need to remove to bring down the total rate of infection? The green box marks what's even possible to do with symptomatic isolation. And so for symptomatic isolation with COVID-19, the green box in each of these graphs in the top row, you can see that even if you isolated people perfectly, at the current transmission rates, it's almost impossible to really remove everyone from the population that you need to slow down the epidemic. In contrast with SARS, it was much easier to accomplish that. So here's an example of sort of how you can use strategy. And so the take homes from this are symptomatic isolation is not good enough. You need to reduce your transmission rate, which is the top left panel for COVID-19 here. And then you can actually have a fighting shot of dealing with this. But a lot of the things that officers are doing in terms of symptomatic isolation end up being hygiene theater, because it's very, very difficult to catch everyone who is infected. And the WHO has said 40% of infections are from asymptomatic people. So here's an example of why this worked well with SARS. It didn't work, and it won't work with COVID-19. Ominously, slide 7, you can do the same comparison with Spanish flu. And what you can see over here is that this disease will be harder to stop than the Spanish flu, because that the space of the green box, as you can see, has more wiggle room for the Spanish flu than it does, marginally more wiggle room for the Spanish flu than it does for SARS-CoV-2. So as I said, here's an example of the kind of things you can do with epidemiological modeling. You can construct heat maps sweeping over different parameters and start asking questions about strategy. The question we asked in this particular couple of slides was, how effective would symptomatic isolation be when you're trying to control COVID-19? And the answer we learned was not very much. But you can use the same lens, the same prism to ask other kinds of questions, which I'm going to now do. So switching gears a little bit, a primary concern with SARS-CoV-2 is the short duration of natural humoral immunity. So this is a recent preprint, one of a number of them. This is the one out of Boston Children's Hospital, but there's one that's come out with from a British group. There've been a number of others out of Germany as well that show short half-lives. And what you can see over here is that on the left-hand side, you see a number of pre-pandemic controls, over 1,500 pre-pandemic controls, for whom IgG levels were assessed. And then on the right, you see about 250 patients for which PCR positive patients for whom antibody levels were assessed over 70 days. And so what the researchers did, it's a really nice piece of work. They had 250 of these patients, they sampled all of them, and then they drew a line with a Sharpie through that. That part was unfortunate. That's called a spline fit, and is not how you assess half-life. Basically, they just put a random polynomial down there, which doesn't tell you very much. So we took that data set and we did what's called a population PK analysis, which is we did the pharmacokinetic modeling for it. We took each patient's pharmacokinetics separately, and we fitted a model to all of the patients together, accounting for inter-individual variability as well. So this is a population pharmacokinetic model fit to the data. And when you look at the pop PK model, the model fit, the standard errors are really good. It's like 5 or 6% RSE, which is really tight. This is called a visual predictive check. The red line in the middle shows you the mean and the uncertainty around the mean. The blue lines are the 95% confidence intervals. And you notice immediately that the 95% confidence intervals are very wide. And that has to do with the high level of inter-individual variability that we're seeing. with each of these patients. So what that means is, even though our model fits the data quite very tightly, in fact, there's quite a lot of noise. There's quite a lot of inter-individual variability. It's not noise, it's biological variability in how people respond to this. And so now we took a look at that and we said, okay, well, if that's the case, now can we build a model to ask the question, how long will it take for individuals to return back to baseline using this data? So you can use this data and simply extrapolate those lines out and say, how long will it take for people to return back to baseline? And we did that. And over here, you see a graph. It looks like a survival curve. It's a percentage of the population that still has IgG levels above baseline. And for IgG levels, the mean is 154 days. So almost everybody comes back to baseline at 1 year. Now, I can hear you thinking, but that's just IgG. What about IgA? What about IgM? Well, we looked at those. Those half-lives are shorter. You could look at the question, well, what about CTLs? Maybe there's T-cell immunity involved. And we've been digging into that. We have a data set that we're modeling right now, and it turns out that T-cell immunity kind of tracks roughly with this as well. You can also ask the question, what happens with affinity maturation? So we did a similar study looking at just neutralizing titers, and the results were basically indistinguishable, meaning although there may be some affinity maturation, it affects things on the order of a few days or a few weeks, not on the order of years. So the notion that neutralizing antibody titer will improve over time and that's going to provide durable immunity isn't really supported right now with the data sets available. So 6 months to get back to baseline, that's really short, but that's not dissimilar from other coronaviruses. So think of this, it's helpful to think of COVID-19 as kind of like the common cold, but it has some percentage chance of killing you. You can catch it as easily, you can catch it as often, but then yes, it does have some percentage chance of killing you. Now, one of the obvious implications of this is that you should be seeing reinfections. And so folks have said, well, where are the reinfections? We don't see them. Well, the proof of reinfections is double sequencing right now. People have said, well, we need to see double sequenced data. So sequence the patient once, sequence the patient on the reinfection, and show us that those two genomes are not related to each other by a simple set of mutations, that it actually has to be a distinct strain. That's a very reasonable standard for proof. If you want to prove reinfection, that's absolutely the way to prove it. And in fact, it bears mentioning, there's a website that has 24 different cases. So that reinfection happens. It's the BNO reinfection tracker is the website. And they have links to the articles and preprints. So that reinfection has been reproven 23 times, so to speak, right now at a molecular level. But you can't use a molecular standard for every single reinfection because essentially almost nobody gets sequenced twice. Very few people get sequenced even once. So we looked at this data set from Qatar, where Qatar had approximately half of its population get infected over the spring and summer. And they actually tracked their, over that period of time, they tracked everybody who was infected, they tracked everybody who was a double positive. In Qatar, they had 250 double positives that were spaced 45 days apart, which is actually quite a high standard. And that had other reasons why they believed that those patients may be plausible candidates for reinfection. So out of the 250 double positives, they went ahead and they only were able to find data for 6, paired sequencing data for 6, and they showed that 4 of those were proven to be reinfections. But if you interpret that data as that there were 4 reinfections in Qatar, you may have missed the point. Because the double positive, many of the double positives that didn't make it to the final cut of testing were very plausibly reinfections. What we did over here is we fit an epidemiological model allowing for reinfection. And we said for varying degrees of protection, how many reinfections would we have expected to have seen in the Qatari data set, allowing for complete loss of protection at 3 months, 6 months, out to 36 months. And the observed number, 256, was in the ballpark of what was actually observed. So if you take the 6-month immunity and you say, well, how many reinfections would we have expected to see? You would have expected to see about 250 double positives by that standard. So this is kind of telling. And what it says is that there may be a lot more double positives that we think are relapses or we think are false positives that may in fact be reinfections. And that having been said, 256 cases out of 133,000 is a really small number. So reinfections are really a small number right now. But notice the shape of those curves. Those are exponential curves. So they're only a small number now. If we have a continued pandemic for a year, year and a half, and this goes on for a while, those reinfections will not be rare. So if you've had COVID-19 already, you should consider yourself in the same bucket as the rest of us, because there's still a good chance that you can get infected. There's a practical consequence to this. So even if reinfection is quote unquote rare right now, exponential curves don't work that way. They change their rate of change, as we all know. So one of the other practical implications of this is that natural herd immunity is out of reach. So we ran a set of epidemiological models positing a different set of R-naughts. And we asked the question, if you look at the graph on the left, what do we expect to see in terms of duration of natural immunity versus the number of infections? And so on the left, what you can see is that if you have natural herd immunity kicking in and lasting for either 6 months, so on and so forth, based on the R-naught, you could have very widely varying numbers of individuals getting the disease. Now, the thing about this is that the R-naught tells you how many people one individual with COVID-19 infects. So the baseline R-naught in the early days of the infection of the pandemic in Wuhan was about 5.7. But there are many countries right now that are at around 1.5, 1.3. But that's not a return to normalcy. No one would say that what we're living through right now is normal. So we explored a range of R-naughts starting from 1.5 to 6, which is the natural R-naught, and said, if we relaxed our controls, if we tried to return to normalcy, we went back to raves and cruises and dinner parties and movie theaters, perhaps not all at the same time, what would happen? And the answer is you would end up with a massive burden, a hellscape of unending disease. And on the right-hand side, you can see what the yearly US deaths would be based on the current infection fatality rates. So it would be a staggering burden. It's not doable. Even at 24 months, it's kind of not doable. So unfortunately, natural herd immunity is out of reach, is not a practical solution. So that brings us to a related topic, which is, what if some people believe in herd immunity and some people don't? So there's this branch of math called game theory. And many of you know about it from maybe A Beautiful Mind or from the story of The Prisoner's Dilemma. If you have two prisoners and each one doesn't know what the other is going to do, their payoffs are different depending on what the other person chooses. And a Nash equilibrium is reached when neither party can make a choice that is better without the other person changing their choice. So in other words, a Nash equilibrium is a no regret situation. So we used game theory to explore the question of what is the incentive to not comply with COVID-19 measures, control measures. And we said, okay, if you take the cost of intervention divided by the cost of infection, and you compare that to the efficacy of an intervention, what happens? And what you can see over here is the red region marks the area where people have an incentive to not comply. The blue region marks the area where individuals have an incentive to comply. And what happens is that individuals will often perceive themselves as being in that red region, that, okay, if I take on these interventions, they're going to cost me a certain amount. And my own cost of getting infected is low, because if I get infected, nothing bad is going to happen to me. That could put you in the red region very easily. So it's entirely rational to not comply if you believe that your personal cost is very low. And what this shows is that there's a shifted externality, that those people who believe that cost of infection is low, or those people who believe that their outcomes are not going to be that bad, can rationally choose not to comply. And so this is where public health messaging is focused a lot on altruism, and altruism can only go so far. One obvious public health implication from this is that public health messaging should emphasize the costs to the individual for COVID-19. Pretty much everybody has some non-zero risk of a really bad outcome with COVID-19. If you read the literature carefully, young, healthy people are getting long-term health effects going with this disease. So it turns out that short-term suppression of COVID-19 requires a high degree of compliance with a highly effective measure. And you're seeing that over here. So what we did is we looked at it, we ran a set of simulations, and we said for different intervention efficacies at different fractions compliant, what would it take from where we are today? Say we had a vaccine, and the vaccine worked at 50%, and the FDA approved it. What percentage of compliance would we need to eradicate the disease? The heat map on the right, you can see in the graph on the left, the heat map is the total number of US infections. So you can see that a modestly effective vaccine with a certain percentage of non-compliance is a non-starter. Even with a 70% effective vaccine, you need greater than 90% of people to take the vaccine in order to have something that actually is bringing the rate down. The right-hand graph is the top left corner of this graph, the larger graph blown up and placed on a log scale, so you can really see what's happening. And you can see that it takes about a 90% effective vaccine with 90% compliance or greater to get fewer than several 100,000 infections a year in the US. So if you set the threshold and say, look, we want to go back to normal, we don't want to wear masks, we don't want to do these things, we just want a vaccine. Well, that vaccine's got to be at least 90% effective, and at least 90% of people have to take it, or something in that ballpark. A 50% vaccine will mean that the pandemic just continues under the current conditions. So this is a long, now when we look at the long term, what you can see is what is the impact of non-compliance, given that the duration of immunity is short. So on the left-hand side is what happens if we have a 50% effective vaccine and everybody starts living their life as normal over the next 15, 20 years. And this is kind of a common cold situation, right? People have studied the common cold where they show that you can get the disease 5 times or 6 times over a period of 25 years quite comfortably. Thank you very much. So in a common cold situation, what happens is there's no vaccine. So it looks like the line in red on the graph on the left. So essentially you catch the disease multiple times over a period of years. Now, on the other hand, if everybody complied and everybody took the vaccine, and let's say it was a 50% effective vaccine, that 50% effective vaccine would knock everybody's rate of catching the disease down to the blue line. But if you had non-compliers and compliers mixed in, and 70% of the population took the vaccine and 30% of the population didn't take the vaccine, you can see that the compliers' outcomes are significantly worse because non-compliers are present. So if we have a 50% effective vaccine and 70% of us roll up our sleeves and take it, the 30% that doesn't take it will essentially make it highly impractical for us to continue living our lives as if we were going back to normal. On the other hand, on the right, if you had a highly effective vaccine, say a 95% effective vaccine, then yeah, it doesn't matter what the folks who are not taking it do, because the folks who take it will be okay. So slide 15 shows you this concept swept over a range of different effectivenesses and compliances. And you can see that the population level impact of interventions in general is highly dependent on compliance. So if you have a 70 or 80% effective vaccine, you need a certain percent of the population to take it, but there are diminishing marginal returns. You don't need a 99% effective vaccine. As long as you have something that's 70% or more, you can probably sort of squeak through as long as you can get a large percentage of the population to take the vaccine. The graph I showed you around why SARS-CoV-2 is hard to eliminate, hard to control, impresses upon us the reasons why eradication in the long term is the only way to go. Because if you don't eradicate the disease, then people are going to get it repeatedly. And if you don't eradicate it, everybody will get COVID at some point, which again is a roll of the dice on your health, on your long-term health, and will create a massive burden of morbidity for the healthcare system to deal with. So slide 16 is kind of this taken one step further. If the more effective the intervention, the more your incentive personally to take it. I'm not going to go into it in a lot of detail, but you get the general concept. And one other sort of small point to make here is if you have a vaccine that doesn't prevent asymptomatic infection, but does protect you from the worst outcomes, this is only a good idea if asymptomatic patients have no long-term health effects and the infection fatality rate is 0. Sweeping across a vaccine that has 0% protection, 0% reduction in transmission, but that protects you to varying degrees, and looking at varying levels of compliance, you can see that this kind of approach, a vaccine that doesn't prevent transmission, is kind of like a really good thing for the virus, because you'll have hundreds of millions of cases. And this is only a good idea if your vaccine actually prevents long-term health effects and drops the infection fatality rate to 0. Even a very small fatality rate or even a very small rate of long-term health effects will make this into a huge public health issue long-term. So one piece of good news in all of this is that you can actually stack interventions. So if you look at the top left corner, this is what happens when you have a vaccine with a certain degree of efficacy, assuming 18 months of immunity in the population, which is more than we have, and an R0 of 5.7, which is the R0 when people return to normalcy. And you can see you can only return to normalcy in this situation if you have a very high percentage of people taking the vaccine. That's the top left corner. But if you had 2 different vaccines and you had people able to take both, so they're kind of competing in the market, but you can actually take both, that would give you a lot more room to play, right? If people independently chose to take one vaccine or the other or both, like say an intranasal and a systemic vaccine, then all of a sudden the two vaccines really help each other and you don't need as much compliance anymore. If you have a passive intervention, such as say HEPA filters on all buildings, and only 20%, it's only 20% effective, bottom left corner, that helps a lot as well. If you had an intranasal prophylactic that works really well, but not a lot of people take it, that could help as well. If you had universal masking, that could help a lot as well. So all of these things can help. Of course, universal masking is not a return to normalcy. So coupling universal masking with a vaccine is only useful for eradication. But you can see over here, if you have a universal masking mandate and a relatively crappy vaccine, you could bring this thing to heel. So that's a key message also to keep in mind, is we're trying to bring this under control and we can't compel everybody to take it. Then we're going to need multiple interventions at the same time for the people who are taking it, who are taking steps to eradicate COVID. One other point to bring up is this question of mutations. So one of the things that has been said is that, look, there's not a lot of potential for mutations, so there's nothing to worry about. What we've done here is we've shown a set of images of published data sets around the spike protein. And what you can see in panel B on top is what the spike protein looks like, front, back, and top views. And the dark red epitopes, the dark red residues, are the ones that show up in a large number of different epitopes for antibodies that have been published. And these are all antibodies that arise naturally in humans. So you can see that the naturally arising antibodies kind of cluster on top of each other on a narrow section of the spike protein. Now, that section of the spike protein, it turns out, bottom panel, has already been mutated experimentally and has been shown to generate escape mutations. That's panel B on the bottom. Panel C shows you that those escape mutations don't actually result in a very large change in ACE2 binding, the cognate receptor for SARS-CoV-2 very often. There are many escape mutations that actually don't impact ACE2 binding at all. And panel D shows you that the prediction accuracy of the degree of ACE2 binding for infectivity, or the prediction accuracy of the degree of expression for ACE2 binding for expression, is very poorly linked. So in other words, the virus can and this is an RC curve that was derived from experimentally published data, experimentally derived data that was published. So what we're showing is that we're showing three things. One is the natural residues all cluster on top of, the natural epitopes all cluster on top of each other in terms of where they bind on the spike RBD. Those epitopes are amenable to generating escape mutations, panel B on the bottom. Those escape mutations don't have a negative impact on ACE2 binding. ACE2 binding itself doesn't predict a whole lot for infectivity or for that matter, expression. And here's the creepiest part of the whole thing. Panel A, the purple figure, that shows you what's been reported in the GISAID database already. The darker the color, the larger the number of variants that have been reported in the GISAID database. The GISAID database is the database of all the sequences of SARS-CoV-2 that have been published. It's a repository. And for a viral sequence to show up there, it has to be not only viable, but viable enough that it was the dominant viral sequence within some patient somewhere. What you're seeing here is that many of the residues in the spike protein that are targeted by antibodies and that are capable of generating escape mutations exist in the GISAID database with multiple different variations, suggesting the existence of neutral genetic drift. Now you can say neutral genetic drift is great. Why do we care? That means that the virus can't mutate. Well, for now, what we know is that viral load peaks within patients around day 0 of the infection. The immune system shows up day 7 or day 14. So it's safe to say that a lot of the transmission right now is happening in the absence of active selection pressure from the immune system. So all of this is happening under drift. And what the virus is showing you under drift, it's tipping its hand. It's telling you, look, we can mutate this residue if we have to. When faced with neutralizing antibodies, the virus will be able to readily generate escape mutations at a low evolutionary cost. This is something that, so this is part of a paper that we're getting out, we're pushing out the door right now, but this is something that bears mentioning, that it's actually the tolerance to mutation that is already visible under conditions of neutral genetic drift. That tells you that when the antibodies show up en masse, whether it be vaccines or neutralizing antibodies as therapies, the virus still has a latent capacity to escape those antibodies quite readily. We then explored this question one step further, and we did evolutionary modeling to ask the question using a technique called mutation selection balance, which was originally used for HIV, to ask the question, well, how frequently would we expect to see single mutants, double mutants, triple mutants, and quadruple mutants, meaning mutants, viral mutants that are capable of defeating one antibody, two antibodies, three antibodies, or a four antibody cocktail. And we looked at a range of different pandemic sizes at the bottom, as you can see, ranging from 5 million to 640 million. For reference, right now, the number of infected individuals worldwide is 50 million. And we said, okay, for different amounts of fitness costs, for different amounts of damage that it does the virus, what can we expect to see? Sorry, can you see my screen?
Yes, it's, yeah, now we can see it.
Okay, so what we did was we swept across a range of different fitness costs and we said, how many mutants can you expect to see? And what you can see over here is single monoclonal antibodies used as therapeutics will have large numbers of individuals infected with viruses that are capable of defeating single monoclonal antibodies. Regeneron's double antibody cocktail is a good idea because if it was used in a prophylactic setting, there's a possibility that that single patients, that individuals might not have those double mutants, but you can see that there'll still be a small number of patients that will have double mutants present. If you wanted to use a triple antibody combination to fight the virus, that is something that begins to work. In other words, the likelihood of having standing genetic variation have enough alone be responsible for enough people in the world having viruses that are simultaneously resistant to 3 antibodies is low. And quadruple antibodies are, that probability is even lower. So either for a vaccine that generates large numbers of different antibodies or a diverse immune response, or for large numbers of monoclonals that are being used prophylactically, there's still a path forward. But using a single antibody or a vaccine that only generates a sort of a very narrowly focused immune response, or double antibodies prophylactically will not be as effective, is what we predict from looking at the evolutionary cost to the virus from escape mutations to the spike protein and coupling that with traditional evolutionary theory. We also looked at the question of what would happen if under evolution. So the previous slide, slide 20, was under neutral genetic drift. This means standing genetic variation present in the population already. But when a treatment shows up or when a prophylactic shows up, it actually provides a positive selection advantage. And we asked the question, sort of what happens now if you have a positive selection advantage? How many months would it take until the mutants were established? So if you hypothetically used a single monoclonal antibody prophylactically across the whole world, how many months would it take? And the answer is it wouldn't work. If you used a double monoclonal antibody, how many months would it take? And the answer is for a pandemic size of about 40 to 80 million, it would be, again, on the almost instantaneous. But when you get to triple and quadruple antibody combinations, the likelihood of those evolving under selection pressure in the presence of an antibody prophylactic are low. So again, as long as your vaccine generates a diverse enough immune response, you're going to be okay. But if your vaccine has a couple of different highly immunodominant epitopes, you may be headed for problems with resistance. That's the take home here. So that's kind of, that summarizes our work, that summarizes what we've been doing. And as I said, I emphasized our work and the modeling we've been doing. But there's plenty of other papers, and we can talk about it, that support a lot of the points that we're making. So we're working pretty much in lockstep with what is being seen. It's a grim view, but there is a silver lining. There is a way to think about this virus, and that is to throw the kitchen sink at it. We should be focusing our public messaging on eradication when it comes to public health. We should be looking at ways to reduce R0 using passive measures, things like HIPAA filters, things like, for example, looking at ways to reduce transmission in congregate settings that don't involve individual choice. As I said, any kind of passive measures that can come up. Prophylaxis should be approached with multiple different measures. So looking at things like vaccines, intranasal prophylactics with antivirals, sustained long-acting monoclonals if possible, doing whatever we can to throw as many different types of things. And for each different type of approach, making sure that the market has multiple different products available, because when the market is dominated by a single product, as you can see from this previous slide, resistance will develop more quickly. So the more diverse the marketplace, the harder the virus will find it to react. And this is a really important point, that people working on COVID-19 shouldn't be looking to maximize market market share, because maximizing market share maximizes selection pressure and speeds the emergence of resistance. So you can think of it as sort of farming your own market share if you're working on COVID-19. And of course, seek to reduce mortality using novel treatments. So the direct implications for the design of therapies and prophylactics are that highly effective prophylaxis is something worth focusing on. Interventions must be stackable, and having highly effective interventions with a low compliance will be helpful, just as modestly effective interventions with high compliance can also help. And so if we have a vaccine that works okay or works so, not so great, but at least reduces transmission dramatically, that could stack quite nicely with other kinds of prophylactics at the same time. And all prophylactics must be designed with resistance in mind. So in other words, having multiple different kinds of therapeutic approaches and prophylactic approaches, and for each of those to have multiple different products on the market at the same time, preferably non-overlapping, is going to be really, really important. When this pandemic started in the spring, we all sort of recognized it for what it was. I think many people did, and kind of like there was this bad feeling in everyone's mind, at the back of everyone's mind, that okay, this is a historic threat, and we mobilized the biomedical community mobilized like never before. And the analogy that was used was the Manhattan Project, and it makes sense, right? The Manhattan Project was the best and brightest all in one room, and they came up with something in a blazingly fast amount of time, blazingly short amount of time, so to speak, and it shocked and awed the Empire of Japan into submission. And it's very easy to cow an enemy government into submission through an act of intellectual brilliance, or through an act of finding a weapon that your opponent has no answer for. But viruses are not like that. Viruses are not impressed, and the more focused we are, the more of a patsy the human race becomes for COVID-19. Because all that it means is that if you throw a single intervention to the virus, that's going to be the best possible thing you can do if you have the virus's best interest in mind. So the Manhattan Project is really the wrong analogy for this, and it's messy, but this is a much better analogy for what we should be doing. We should be trying to get as many different things out there, and we should be trying to use them in parallel. There's a lot of talk about how we should focus and how the marketplace should not be allowed to decide. Well, it's not really the marketplace that will decide. It's the virus that will decide. And having committees of people decide which interventions are worth putting all our chips on is probably not the best idea. If the committee is not the final deciding authority, if it is the virus that is the final deciding authority on what it will take to control it. So that concludes my talk. I wanted to acknowledge the team. There's a fairly large team. We have multiple active collaborations with labs in BU, MIT, and Harvard right now, focused on different aspects of this. And I wanted to acknowledge all the folks who've been involved. We do have, as I said, a set of therapeutics that we are designing with these principles in mind, and we are working on those. But a lot of that stuff is currently not ready for public disclosure. And with that, I'll take any questions. Thank you.
Thank you so much, Arijit, for your presentation. And I saw you mentioned Diane Joseph McCarthy. She is in our room watching. I believe she's the executive director from Boston University. And, yeah, and there's just one quick question for you from James Keats, Chief Scientific Officer. In one of your slides, he said, what is missing here is the change in IgG and T cell amnestic response upon re-exposure. Could the lower number of reinfections point to this memory response?
So that's a great question. It could. There's another explanation for the lower number of reinfections, and that is it's an exponential process. So reinfections should be living on an exponential curve. What I showed, assuming a 6-month reinfection, a 6-month duration of immunity, what I showed with the Qatari data was that in the Qatari data, you should have only seen a few hundred reinfections at all. So you should only have seen about 250 reinfections, which is bang at the number that they had for the double positives. So reinfections are currently rare, and they should be rare because because 6 months immunity. But give it a year, and they won't be quite so rare. So we don't know yet whether there is a strong T cell component that could kick in. It's too early to say. So that formally is possible. But again, the lower number of reinfections should not be interpreted as indicative of other immune mechanisms until we've waited long enough to see that low number remains low.
Okay, and there's another question from Dr. Jonathan Davis, who just presented, Head of Innovation Discovery. He's asking, regarding reinfections, is it likely that they would be lower severity given the presence of immunologic memory?
Yeah, that would be nice, right? That would be really nice if it were true. So far, the data doesn't suggest that. So again, if you look at the reinfections, we ran a sort of a little bit of statistics on it. We looked at sort of from less severe to more severe, first infection less severe, second infection more severe, or vice versa, what happens. And so the numbers are small right now. There are only about 17 cases. And I think there were 10 that got worse and 7 that got better. So there's no statistically significant p-value on that one. In fact, one person got reinfected the first time in Holland with a mild infection, and the second infection killed her. So we don't know yet. But that individual had a compromised immune response. So what I'd say is we don't know yet. We do know that people have had more severe reinfections the second time around. One piece of good news is that there's probably not antibody-dependent enhancement, because if there was, the reinfections would have been uniformly more severe, which they are not.
Wonderful. Awesome. Thank you so much, Arijit, for presenting. And if you're interested in asking more questions, do let us know. We can connect you with him directly. Thank you.
Yeah, and that deck is for sharing. So, please feel free. If you have questions, you can ping me on LinkedIn. Again, we see this as being very much a sort of public service thing, and really sort of having the conversation pre-competitively with everyone around this, because, as the cliche goes, we really are all in this together.
Indeed. Appreciate having you, Arijit.